Thank you guys very much for taking a little time out of your day. All right, hang on a second. Apologies. I'm having some minor technical issues I'm going to see if I can solve. Uh, um, mhm. Okay. I'm going to try stopping that for a second. I'm going to try resuming that. Um, that's a bummer. Okay, I can soldier on. The Zoom is covering part of my slides, making it a bit challenging for me to see my own presentation, but that's okay. So thank you very much for taking the time out of your day to come here this evening and hear a little bit about tawny crazy ants. And I'm going to be telling you, I can't see my title, so I won't say exactly what I'm going to be telling you about, but I'm going to be telling you about some work I've been doing since starting back in 2009 on tawny crazy ants in Texas, and then also work I've been doing in South America on the same species in their native range in Argentina, and then work on a pathogen of these ants that we found. Okay. Okay, so tawny crazy ants are from northern Argentina, southern Brazil, Paraguay, Uruguay. This is the same part of the world that imported fire ants are from, and also Argentine ants. And you may not realize, but Argentine ants actually invaded the southeastern United States back in 1891. And were widespread until the introduction of imported fire ants in the 30s and 40s. Imported fire ants largely displaced Argentine ants, which are now restricted to some woodlands and floodplain areas of the southeastern U.S. Tawny crazy ants were introduced into South Florida probably back in around 1990. It's not really clear when they got into Florida because there's a very similar introduced ant in Florida that they were confused with for a long time. But they then rapidly began to spread to other counties in Florida, were introduced into Texas in 2002, and have since become established in all of the states in the Gulf region. Now, these are county level maps, so that just means there's one population in one of these counties, and obviously the map's a little out of date now. It does not imply that the entire county is covered in tawny crazy ants. Their distribution, their potential distribution, I collaborated with a fellow to do a environmental niche modeling, which basically took occurrence records from South America and from North America and used all the environmental niche envelope, all the temperatures and humidities and first frosts and information from those environments, and constructed what their potential niche in the United States would be. And this is what you're looking at here. Essentially, orange and yellow areas are high probability of suitable environments for them. And you can see that that's fairly reflective of where they're already established. It doesn't seem like they're going to be a problem outside of the southeastern U.S., outside of the Gulf State region. These ants are omnivorous. They feed on both vegetable and animal matter. They scavenge and prey on insects, and they feed a lot on honeydew. So that's the sugary secretions of aphids and homopterans of all kinds, scale insects, membracids that jump hoppers. they're ecologically dominant, which means that they are really good at monopolizing the food resource base in the environments that they invade, and they successfully displace other ants from those resources, and other insects. They have multiple queens per nest, sometimes hundreds of queens per nest, and they nest opportunistically, which so they don't dig extensive mounds or nests in the earth like a lot of ants. Instead, they occupy empty cavities either in the soil or in rotting wood or vegetation, and they move a lot between these cavities. This slide's a little incorrect. They do not engage in alate flights, but males do fly. Females do not engage in alate flights. And apparently that all the mating that occurs is within the nest, and that spread is then through nest fission. So a big nest, small nest goes into a big one, and then it divides into two. What this means is that because they don't have alate flights, they can't found independently. So all the populations that occur in the United States occur because somebody moved the ants and deposited them somewhere new, either in a potted plant or some trash or whatever. And then from that point of introduction, they expand radially out up to about 200 meters a year. So on the lower left, here you can see the edge of the expanding population over a 3-year time frame, and the point of introduction was this little pin back here. So it leads to a radial plaque across the landscape. These populations can become extremely dense, and such that the entire ground is just seething with ants everywhere you go. And this typically occurs late in the year, in the late summer or fall. Now, they don't do as well in drought years as other years. So this year has actually been, relatively speaking, a lot lower abundance than some years. And then in the spring, after winter, they come out at much lower densities, and it might not be till late summer that the population densities are high enough that they are really problematic. Depends on the year. They have all kinds of impacts on people. So they move into electrical circuit boxes, and they short out the circuits because the electrical boxes have the sort of cavities they need for nesting. And they move into people's houses in huge numbers, and they put down, barrier pesticides to try and keep them out. And you end up with these huge drifts of dead ants around your property, around your house. They tend underground root homoptera, which are aphids, and above ground homoptera. So this is a variety of things they're tending for sucrose. Well, this has impacts on agriculture because these sap-feeding insects damage crops, and so they can cause agricultural damage that way. They can also destroy honeybee hives, as Gary was mentioning earlier. And finally, they'll directly attack vertebrates like this calf when they're in the very, when they're just born and their eyes are opening. This calf has got damage to its eyes as a result of crazy ants. They also have really substantial ecological impacts, which is more what I study and what I'll talk a little bit about. They reduce the species richness of co-occurring ants, and this species loss is non-random. So interestingly, large-bodied species are displaced or are eliminated from the system, large ants, and very small ants are not impacted. And this is probably because the large-bodied ants are essentially, building colonies with tunnels that are of a large enough diameter that the crazy ants can use them, and they're just kicking these ants out of their nests and taking them for themselves. This plot shows whether or not a species was negatively impacted, essentially eliminated by tawny crazy ants, or whether it was unimpacted. And then on the x-axis is the head width of the largest ants in the colony. And so the large ones are negatively impacted and the small ones are not. It's also divided up by where the ants are from. So tramp species that move with human commerce are tend to be very small species of ants. And so what happens is that after these crazy ants invade, the ant community you're left with is all these tramps from all over the world, these globally distributed species, and all the native Texas species are largely gone. These impacts aren't just ants. These impacts are widely distributed across arthropods generally. So arthropods being insects, spiders, other kinds of arachnids like scorpions, and isopods. And you can see these are very, in these plots, what you're looking at is, in the upper left here, this is the field site, and these plots were done structured over space. So behind this line is areas that were invaded by tawny crazy ants, and the farther you get, this dotted line is the edge of the expanding tawny crazy ant population, and the more negative the value, the farther behind the tawny crazy ant front you are, and the longer tawny crazy ants have been present at that point in space. And then in front of the line is habitat invaded by imported fire ants, which is pretty much all of the open environments in Texas. And you see this very significant depression of arthropod abundance generally, very, very severe. And that's sort of all arthropods. And you see similar kinds of impacts to species richness that I was showing with the ants in the arthropod fauna generally as well. Not as severe as abundance. You still have quite a bit of richness, but it's very few individuals. And that just means the woods and the prairies are just very quiet. There's no insect sound, there's no bird noise because there's nothing to feed on. And birds and wildlife generally are also impacted, so that ground nesting vertebrates are very scarce in these areas, particularly lizards and snakes and rabbits seem to suffer quite a bit. The one good thing that these ants do is they very rapidly displace imported fire ants. So this is the same plot I was showing you a minute ago. This is the same site. This is actually an Iowa colony. The areas on the x-axis, again, these are areas invaded by tawny crazy ants, and the farther back you go, the longer it's been invaded. And you can see on the upper plot here, this is the abundance of tawny crazy ants. And these are pitfall traps that are set out for about 24 hours, they're set out for exactly 24 hours. And they are not invaded. It's just a small tube in the ground with some soapy water in it. And you get thousands of ants per trap in these 24-hour periods in these high density areas of tawny crazy ants. And then in front of the front, there are no tawny crazy ants. And as you see, imported fire ants are essentially eliminated behind the front and abundant in front. This shows the same thing, but just in a plot followed over time, so that you see the annual cycles in tawny crazy ant abundance and the disappearance of imported fire ants and other ants from these plots. So how is it that tawny crazy ants can eliminate such imported fire ants? Everybody knows imported fire ants are tough customers, and they are tough customers primarily because of their venom. Their venom is, we think of it as painful, and it is, but In fact, 98% of their venom is an alkaloid, or a group of 5 different alkaloids that we don't feel. The pain component is a very small part of it, which are proteins. The alkaloids are actually insecticidal, and they fire ants use these to prey on other insects and also to fight ants for food items. So they essentially will use their stinger like a paintbrush, and they'll smear their venom on competing ants, and they will die. So one reason that tawny crazy ants could be displacing imported fire ants is just numbers. For some reason, they're able to achieve these incredible densities, and as a result of those numbers, they're able to overwhelm fire ants. That's not the case. So when you control for the abundance of the 2 species, so if you go across this boundary of expanding crazy ants and you find the sweet spot where their densities are exactly the same, and you look at how competition plays out in those environments, what you see is that tawny crazy ants are slightly are behaviorally dominant. They win interactions with fire ants even at equal numbers. So on the x-axis here, this is the relative abundance of the 2 species at a given spot. And then on the y-axis is the probability from 0 to 1, whether or not of tawny crazy ants winning an interaction. And what you see when they are absolutely equally abundant, there's about a 68% chance that tawny crazy ants will win the interaction. And when fire ants, when there's a 50% shot of either species winning, if it's going to be a coin toss, then fire ants have to have a 2 to 1 advantage in numerical dominance, numerical abundance. So how do they do this trick? And this is how, and I'm going to show you a video here. And what you're going to see is this tawny crazy ant here. She's spraying venom at this fire ant, and then she goes up and she takes the droplet of venom off. So I'm going to back that up. So what you're going to see here, let me just back that all the way up. We'll start over for just a second. This ant here is a tawny crazy ant. This ant here is a fire ant. You can see it's the fire ant stinger here. You're going to see the fire ant exude a droplet of venom to smear on this attacking crazy ant. You're also going to see the crazy ant spray the fire ant with venom. And then what I want you to see is what happens after the crazy ant gets the venom. So the crazy ant sprays the fire ant with venom, goes up, actually takes the droplet of venom off the fire ant stinger. That's suicide, should be suicide. And then she goes off, and then this behavior here is what I want you to see. She's going to tuck her abdomen under her body in just a second. And she is, there, we're waiting for it, waiting for ants to behave. There. So she tucks her abdomen under her, and she takes something from the tip of her stinger. And then she goes up and she's going to take that venom droplet again. Let's see, there's the behavior. She goes up and she once again sort of disarms the fire ant by taking the venom droplet on her mandibles. Okay, here's the behavior up close. So this is me applying venom to this tawny crazy ant. So she's grooming, and then she takes fluid from the tip of her acetopore, her venom gland, and she spreads that fluid all over the area that was exposed to fire ant venom. So this looks for all the world like this ant is trying to detoxify the fire ant venom. And so, and that in fact is what's occurring. So to work that out, what I did was I covered up the acetopore of tawny crazy ants with nail polish, shown here, and then on control ants, I did the same thing. I put a little bit of nail polish right on the top of their abdomen. And then I exposed them to fire ants and waited until the fire ants hit them with venom, and then pulled them aside and watched, how well they survived. And ants, the crazy ants that did not have their acidopore sealed were survived 100% exposure to fire ant venom, where those that had this treatment of having their acidopore sealed died. And it turns out that you can replicate this result with the compounds involved. So there's a couple of different things in tawny crazy ants' abdomens that could be responsible, but it turns out it's their venom. It's formic acid, which is what tawny crazy ants use for venom. And it completely detoxifies the alkaloidal components of the piperidine alkaloids that are in fire ant venom. So, so I showed you that they are at these really extreme densities. And here's just another example of that. That's a tree. This is a different site in South Texas. And this is a tree. And these columns of ants are all around the tree. The whole tree looks like this. And every tree in the woodland looked like this at this time. This is at Estero Llano Grande State Park in Weslaco, Texas, in 2006, I believe. So how do they achieve these extreme densities? No, 2016, apologies. One answer is their social organization. So tawny crazy ants are what are known as supercolonial. So and what that means is there are no boundaries between nests within these populations. So these areas that are infested by tawny crazy ants, which can be incredibly expansive, they can be many kilometers in diameter, and within that circle, everything in it is full of tawny crazy ants. All those ants are members of the same colony. They're occupying different nests. They're moving between nests. This kind of social organization has, some ecological benefits. It has a distributed foraging network and reduced competition, but intraspecifically, so tawny crazy ants aren't competing with each other. And it allows them also to integrate functionally across space, so they can, where they can move and concentrate in areas where there are high resources and where life is good, like as the woods, for example, in times of drought, and then they can move back out into the prairie when it's wet and there's lots of food in the prairie. There are some possible ecological costs to this, which is an increasing vulnerability to pathogens. And this is a hypothesis more than a demonstrated fact. It's something that's been speculated about because these societies are so open. They're changing workers between nests. They're engaging in trophallaxis, which is regurgitative food sharing. It's how ants share food amongst colony members. And all these food and workers are just flowing over between nests. There's no boundaries to pathogen spread. So we know that that tawny crazy ants lack behavioral boundaries between nests within populations in Texas. So anywhere within a population, like in an individual infestation, there's no boundaries between nests. But what about in the native range? And what about between populations in the southeastern US? And since we don't see intraspecific aggression in these ants, what does it look like? Do they even have it? So we did a study where we looked at what intraspecific aggression looks like across populations within the southeastern United States, and also across populations within Argentina in their native range, across and within. And we did it in this what's called a neutral arena assay. So it just means that we take a little vial, we take 5 ants from one colony or one collection site, we put it in the vial, and then we take 5 ants from another and we put it in the vial and we see what happens. How do they behave? What do they do? Do they fight? Do they not fight? What do the interactions look like? And we put it on an aggression scale. So, and this is the scale. Essentially, all you really need to know is that until you get to a score of 3, which is biting, you really aren't seeing aggression. and below that you get things like trophallaxis or allogrooming or occasionally a bite and release, but that's very hard to interpret as to what's going on. So hang on. So in South America, we did this work, and what we found was this, that they would fight in South America. So this is an example of fighting in South America, but it's not sort of full-on fighting you see commonly in other ants. Instead, it's this biting behavior where they curl their gaster tip and they point it at their opponent, but they typically don't spray acid. So this is what's called a stereotypical aggressive display. They don't escalate to lethal fighting. It's stereotyped, these interactions, but they're still really obvious colony boundaries. These ants aren't cooperating with one another. And in the introduced range, so this is these plots, this is the interaction score, average, median. It's the most common interaction score in the US. And these are for nest mates. So you're just taking ants from the same nest, separating and putting them together. That's what you get. That's how nest mates treat each other at those kinds of interaction scores. Within the same population, about 50 meters apart, this is the same. You get very similar results. Between populations, so this is like comparing ants you collected in Austin to ants you collected in Houston, and you get the same result. And then from different regions, so ants collected from Florida compared to ants collected in Texas, and you get effectively the exact same result. And this dotted line up here again is about where you start to think, okay, these ants are actually being aggressive to one another. So throughout the southeastern United States, all ants treat each other as colony mates, as nest mates. Not only are these infestations, individual infestations, super colonies, they're all members of the same super colony that has been transported to other parts of the United States. And this contrasts really dramatically with what you see in Argentina. Nest mates, the interactions look the same as you might expect. And then as soon as you get outside of nest mates, you immediately start seeing aggressive interactions. And if you're talking about distant pairings, which are about 100, this is about 100 kilometers apart, you never see anything but aggressive interactions. You do see some, there's some close pairings where you see friendly behavior, you see a range in aggression, and that's because there are some expansive colonies in South America, just like there are here, but they're nowhere near as large. So Nylundura fulva is unicolonial in the southeastern U.S. That just means it's just one colony, one giant super colony, but exhibits aggression in its native range over small spatial scales. But it's stereotyped aggression. It doesn't lead to injury. And the colony size variation in Argentina is really broad. You go from very small colonies that appear to just be a single nest, as far as you can tell, to areas of about 100 meters in diameter where every nest is cooperating and members of the same colony, but never saw anything larger than that. So as a consequence of introduction in North America, Tawny crazy ants have transitioned from this multi-colonial to a super-colonial state. And that's probably because only one colony was introduced. And this is the most parsimonious answer. The simplest answer is that only one colony was introduced, and it has kind of expanded without constraint and been moved around. This slide's a little out of date. There is now genetic data that supports that hypothesis. There's very little genetic variation between populations of tawny crazy ants within the southeastern United States. Okay, so this is essentially what I just talked. Am I going the wrong direction? Okay. Okay, so we were moving along doing this work, trying to understand more about this invasive ant and making progress. And all the while, and then we had an unexpected discovery. We found some ants that actually ants from Florida that had these very white swollen abdomens. So this is a picture of these white swollen abdomens, and that's a symptom of can be a symptom of infection by a microsporidian pathogen, which I'll tell you more about. So we ground them up and we looked at them under the microscope, and sure enough, we saw these spores inside the ants. We took the spores out, got the spores, did some molecular biology, ground them up, ran some PCR reactions, and figured out that managed to get DNA sequence data back, and figured out that this was an unknown microsporidian. It wasn't even related to any other microsporidian that was known to exist. And so we described it, and it's a new genus, a new species. It's called Myrmecomorba nylandaria. And it's the first documented pathogen of tawny crazy ants. We found there's four spore types, and spores are found in all castes. Well, actually, sorry, I keep finding these out-of-date slides. They're not found much. They're found only infrequently in queens, and I think those are due to environmental contamination. So they're found in all castes but queens, and they're known from scattered populations in Florida and Texas. We do not know where this thing came from. So what are microsporidians? I talk a little bit more about them, but microsporidians are essentially they're related to fungi, and they are intracellular pathogens, which means that they complete their entire life cycle inside the cell of their host. They form a spore, and their transmission is between cells. It's kind of a virus-like biology where they have this coiled polar filament inside of the spore. And then when their spore meets the conditions for germination, which occur in the midgut, in the gut of the host. So when they encounter, and they're sort of near epithelial gut cells, through osmotic pressure, they fire out this polar filament. It punctures the cell wall of the host, and then they shoot the entire contents of the spore through this tube and into their host cell. And then once inside the host cell, they sort of hijack the host cell's machinery of self-replication and use it to replicate themselves, replicate more spores. And in the process, when they get into a fat cell, they proliferate really wildly, and they hijack it and form these sporocystic sacs, which is those white swollen gasters abdomens that I showed you a while ago. I told you that they are related to fungi. Some people call them fungi. Some people say related to fungi. This is the latest data we have on it. So this is a phylogeny. Phylogeny is just a diagram of genetic relatedness between organisms. So the shorter the branch length between two organisms, the more closely related they are, the fewer genetic differences exist between the two species. And this group is all of what's commonly known as fungi. And then here are microsporidians sitting well outside, really. And so Microsporidia plus Rosalida should probably be their own kingdom of life, but that's not currently how they're defined. So we just say they're related to fungi. They're an ancient group of life, and they're extremely diverse. And honestly, we don't know a lot about them. They tend to attack insects and fish, and so that's where we know the majority of our diversity from. This phylogeny is all the known, well, not all the known Microsporidia by any stretch. It's representative taxa of known Microsporidia. And the blue arrows are other species of Microsporidia that attack social insects, so either fire ants or thief ants. And you can see there's not very many that are known that attack social insects. And then the red arrow here is tawny crazy ants. And the point is that there's not a lot of signal in terms of host species. It's not like all the ones that attack ants or all the ones that attack ants and bees are each other's closest relatives. They're distributed across microsporidia, and tawny crazy ants are from this very strange little clade here called the Caudiospora. I mean, sorry, Myrmecomorba. So as I mentioned in passing, we don't know where this disease came from. We've found it now in populations all through Texas and Florida, and there's no evidence of a North American origin. We've tested ants, a bunch of ants from North America, and haven't been able to find it. Other insects from North America haven't been able to find it. And there's also no evidence of it occurring in their native range in South America. So we've sampled over 200 colonies from northern Argentina and Uruguay, and tested them for this microsporidian, and we haven't been able to find it. So we don't know where it came from, and we also don't know how it colonizes uninfected populations, although we know it does. So we'll see. As I'll talk about in a minute, one of the things we've been doing since 2009 is these longitudinal studies where we go back year after year and look at the densities of tawny crazy ants in a population, and we will find an uninfected population that all of a sudden the disease is present in. So what do we know? Well, we've worked out quite a bit of other information about this disease, some of which that's pretty important is its transmission biology. So, which is one of the first things we did in the laboratory, was try to understand this. So, the dotted, this is all the different, and this is a study of intra-colony transmission within the colony of tawny crazy ants. And that's really important in this species because, as I said, they're super colonial, which means that within colony transmission is equivalent to within population transmission. These arrows, these represent all the possible pathways by which the disease could move. Blue arrows would be genuine transmission between individuals, and gray arrows are just developmental transitions. For example, if eggs had spores and were infected, and they would then develop into infected larvae, which would develop into affected pupae, which would develop into affected adults. So that's one possibility. We tested all these pathways by in a variety of ways. First, we figured out that queens in nature are not infected, which was a surprise because we had seen spores in queens in our initial description paper. But when we actually did a more comprehensive survey, we found that those spores were very rare and probably due to contamination from the queen just living in an infected colony. And they aren't proliferating in their cells. So that eliminates most of the transmission pathways right there. And then we tested the remaining pathways by essentially creating colonies in the laboratory where you had just infected workers, an uninfected brood, an uninfected queen, or just infected brood and uninfected workers, and a variety of other possibilities. And it turns out that the only way this disease seems to transmit is when infected workers are taking care of uninfected developing larvae. So when that occurs, then the larvae acquire the disease and then develop into infected workers. And so the disease is essentially cycling in the worker population. The next thing we tried to understand was, what does this disease do to these colonies in the laboratory? And we did it under two conditions. We did it under a condition where the colonies had plenty of access to carbohydrates, to sugar, which is, as I said, a really important food resource for these ants, and under conditions where the ants were stressed for sugar. They had very little carbohydrates, symbolized by CHO here, carbohydrates. And we looked at growth and mortality. And what we found was that when they had sufficient carbohydrates, there was a overall impact on colony size, although we didn't really see a significant result in either the growth or the mortality, just trends as you would in the direction you would expect, but a significant result on colony size. And then when we look under carbohydrate stress condition, there were significant impacts on growth, very little growth in the infected fragments, and no growth, in fact, no production of brood. And a higher mortality rate as well, although I guess that was non-significant. But we couldn't look at we couldn't compare overall colony size changes because the colonies just crashed and died too quickly in these conditions. We also found a reduction in worker lifespan for infected workers, a fairly reasonably substantial reduction. And so this disease impacts crazy ant colonies by reducing larval development success and shortening worker lifespan, and it's greater under carbohydrate stress conditions. Now, carbohydrate stress is important because that represents wintertime and that represents drought. Those are the 2 times a year, two conditions under which ant colonies do not have access to sugar, and it happens every year. Okay, now that we knew what we were looking at an impactful disease. We could see it impacting colony fragments in the laboratory. We were wanting to understand what it does in the field, what does it do with field populations. But before going down the road of doing an experimental inoculation, we wanted to know more about the specificity of this disease. Now, so I misspoke a little. We actually went ahead and did the experimental inoculation, not knowing a lot about the specificity of this disease. And we did it because it was already out in nature, right? It's already out there. It's already spreading between tawny crazy ant populations. So we weren't fundamentally changing the system. But because it's important to understand specificity generally, we have done work on specificity of the disease. Specificity is the range of organisms that a pathogen attacks. Or a parasite. So you can have parasites or pathogens that attack a lot of different organisms, and those tend to have really broad ecological impacts apart from just the species of interest. And you can have, and more commonly, frankly, are much more highly specialized, especially in pathogens, highly specialized organisms that only attack one or very few closely related organisms. In the case of this microsporidian disease, we have not been able to find it attacking anything outside of tawny crazy ants. And the way we've primarily looked at this is in examining areas where either you've got native ants interacting with infected tawny crazy ants at the edges of infected populations, or in populations where the tawny crazy ants have receded in the areas where the native ants are recolonizing. And so there's no evidence of infection in ants other than tawny crazy ants. So given that these impacts in the lab, what's going on in the field? What's happening at the population level? And as I mentioned earlier, supercolonial ants are thought to be vulnerable to pathogen acquisition and therefore potentially more able to tolerate pathogen infection, which is a different, separate hypothesis. So we wanted to look at whether this was true for tawny crazy ants, since we had a pathogen impacting a supercolonial ant. So to look at this question, we took 2 approaches. The first was a longitudinal approach where we followed populations through time. And we did that by every fall installing these pitfall traps here. So you can see right here, that little tiny circle, that's a tube in the ground. That's what, when I showed you those abundance data, that's what was actually collecting those ants. And in addition to pitfall trapping and quantifying the abundance of tawny crazy ants every year, we also performed analyses to look at how intensely these ants were infected, how many spores did they have, and those sorts of things. And we did it for 12 widely scattered sites shown here. And in the end, we ended up with 52 year-to-year transitions in tawny crazy ants abundance. So that's like fall of 2000 to fall of 2001. What's the change in tawny crazy ant abundance for that period of time at that site? And what we found was a really dramatic decline in abundance in infected populations. So on the y-axis here, this is the abundance of tawny crazy ants per pitfall trap. And on the x-axis is the number of years since this microsporidian disease colonized a site. And what the general trend here is that for unmanipulated sites, just sites where we're watching what's happening over a period of about 4 to 7 years, populations were going from exceedingly dense, so that's 10,000 tawny crazy ants in one pitfall trap on average for a 24-hour period. So that's just the ground is just a carpet of crazy ants. Going from those kinds of abundances to local extinction, where we can no longer find the crazy ants at these sites, despite looking quite intensively. Now, uninfected populations over that same time period did not decline. So that's this inset plot here. Abundances changed a lot year to year, were highly variable, drought years or a lot of reasons for variation, but there was no overall decline, and they certainly never disappeared. Okay, so the next thing we really wanted to know is, well, can we experimentally induce this population decline? Can we infect populations and have them decline? And so we started this process back in 2017, and we chose 2 inoculation sites. One was Cerro Grande State Park. That was that video I showed you earlier of the tree that was just a column of ants. And the other was a site, a park, Greenbelt here in Austin. One site was, we monitored the ants over an area of 91 hectares and the other over 12. And prior to inoculation, both sites tested negative for the disease. However, at the Estero site, when we were at the point of inoculation, we were ramping things up. We did find very low level intensity infection that had gotten there on its own. So how do we move these disease around? So how do we do these inoculations? And it's pretty simple, although unfortunately not as easy as it's going to sound at these 2 sites, because we were very lucky at these 2 sites. But you collect infected nests, and then you bring those workers in the lab, you pull them, you remove them from their nesting material, test them for whether or not they're infected or not, and what their disease levels are, and then you introduce them into the uninfected. So you find colonies, nests in the uninfected site you want to transmit the disease to. You open those nests up, you introduce the infected ants, and there you go, or potentially there you go. And the reason this works is that social biology I was telling you about earlier, right? So normally in other ants, this would never work because the ants would kill each other, and that'd be that. But because these are supercolonial ants, where there are no behavioral boundaries between nests, either within populations or between populations across the Southeast, we can take ants that are from Houston and we can introduce them into populations in Austin, and those ants will be adopted into the nests and have the potential to transmit the disease. So what happened? First thing that happened over here is that pathogen prevalence increased dramatically, and it increased over from the point of inoculation to at the first site to universal prevalence, and at the second site to very high levels of prevalence within about a year. No less than a year, about 200 days. Disease, and that's across 91 hectares in one case and 12 hectares in the other. So disease spread was just really fast. And then disease burden, which is the infection intensity, how many spores per ant do we see, increases similarly and has kind of a similar trajectory, increases a little more slowly, but you get, after about a year, you're getting up to close to the highest level of disease burden that we see. And then tawny crazy ant abundance drops, craters. So at both sites, we had essentially after 2 years of observation, tawny crazy ants had gone extinct locally at both sites where we did these inoculations. So there's now no more crazy ants at Asteriana Grande State Park, which was really gratifying and a huge success. And also at these Convict Hill site, which is the screen belt. So why does this happen? So the pathogens should not cause the local extinction of their host. That's not how nature works, right? If you cause the local extinction of the host that you're specialized to, you go extinct yourself. So it's it's a bad idea. So what actually is going on here, though, is that within an annual cycle, tawny crazy ants stop producing brood in September. And they do that whether they're infected or not. The queens simply don't lay eggs beginning right about now. And then they begin again laying eggs in March-April timeframe. So there's a 7-month gap in brood production. Well, it turns out when you collect nests from the field and you bring them into the lab, the colony fragments decline rapidly if you bring them in the fall, and they die out in about a 4-month time period. And they die out actually much faster when you bring them in the fall than when you bring them in the spring, summer, which is an interesting effect. And it probably is again related to the cyclical annual production of brood, such that ants you're bringing in the fall are just much older, and infected ants are at the very close to the end of their lifespan. So I call this the mind the gap hypothesis. It's basically essentially that nests don't survive over the winter because this gap in brood production is longer than the lifespan of an infected worker. And so that in the springtime, queens emerge and when there's no more workers to take care of them. And then the reason this leads to the collapse of the entire super colony is because it is a super colony. All these nests are interconnected, and the disease spreads so rapidly that they all go down together. And so this isn't actually a whole population of ants dying out because of the disease. It's one colony dying out because of the disease. It just happens to be that this colony is kilometers in extent. So the answer to that hypothesis about whether supercoloniality can make ants vulnerable is yes. It makes them uniquely vulnerable to pathogens, and that despite scale, supercolonial populations, ant populations, are truly just single colonies, and they have, like any single organism, a shared fate. So thanks. That was mainly what I wanted to tell you about. I'd like to thank my collaborators in this, which are Larry Gilbert, Rob Plows, Pat Folgright down in Argentina, Nathan Jones, Martin Balassi in Uruguay, Melissa Jones, and Todd Bayless. Many technicians over the years that have worked on this project, and lots of support from funding sources. Primarily the Lee and Ramona Bass Foundation have been hugely influential in just supporting our group at BFL over the years. Now, okay, so that's it. So I'm gonna go ahead and I guess we can talk now, and I'm very happy to take questions. So yeah, let me open it up to questions.
That was just absolutely hugely fascinating. We have some in the chat box, but I was writing down stuff quicker than I could even put it in the chat box. Thank you. It's very good. Maybe to kick us off here, just a broad question. Okay, the microsporidia, we don't know where they came from or exactly what they are, but they seem to be unique here. It seems like we have a good history of introducing some pathogen or something in some population to try to control that population, and then what we introduce mutates or goes into a different species. Any chance here we can introduce a greater harm by trying to introduce a pathogen into tawny ants that actually mutates and goes to something else, whatever.
Yeah, so that's the primary concern with biological control generally. And in fact, there's a lot of horror stories out there about biological control. They almost all have to do with people who aren't doing the work of testing specificity. Now, I admitted we didn't do the work of testing specificity a while ago first, but that's because we didn't introduce this pathogen to North America, right? So the problem comes in when you bring a pathogen from the native range of the invasive species and you bring it into an environment where it doesn't exist, and you don't test it to find out if it's specialized to that host. This disease was already here and moving around on its own, so we weren't changing the system by moving it from one part of Texas to another because it was already there. So we didn't have strong concerns about that environmentally. But because it is a cornerstone of biological control to do specificity testing, we went ahead and did that work and are continuing to do it, because it turns out this disease is now, this ant is now invading Costa Rica. And unfortunately from Texas. Somebody brought some farm equipment from Houston. And to our knowledge, this disease doesn't occur in Costa Rica. So before we take this disease anywhere where it doesn't already occur, we want to know a lot more about what its specificity is. Because if a pathogen only attacks the host, then your possibility of other damage is really limited. The changing through mutation is a very slow, very, very rare process. Host shifting, it can happen. You can never rule it out, but it's on sort of ecological timescales. It's unusual. It's very unusual. And the specificity testing we've been able to do to date says it's highly specialized to tawny crazy ants. We haven't been able to find it in other ants or other insects. And in the laboratory, I didn't show you this data, but we've been trying to artificially transmit it to other ant species, and we haven't been able to pull that off either. So it seems like it's highly specialized. Now, this is a bit of a quandary since, again, as I said, we can't find it in its native range, or at least we haven't so far. So how do you have a specialized pathogen that we can't? So maybe it does occur in its native range. There's just certainly something we got to learn here.
Edward,
yeah,
I had a question. So some of the things that we see in South America is there is kind of competition, intraspecific competition. And the question I had asked previously, you'd answered. It seems like it's near clonal in terms of genetic diversity. But what would happen? The question I had asked on the chat was, what would happen if you introduce new males to perhaps create some more diversity? Would that create intraspecific competition that may, in kind of an integrated pest management, stress the ants so that the Sporidia can also be amplified?
Yeah, so that's it's an interesting suggestion. It is actually technically very challenging, and it does create its own risks. So the fact that they are extremely closely related to one another, essentially they have the same relatedness that all of the ants in North America have the same level of relatedness of a single nest in South America. Is a vulnerability for them, right? Because they don't have the genetic diversity to cope with diseases like this pathogen, right? And they don't have the colony diversity to prevent the spread of diseases. So you're going to be, if you were to succeed in that effort, you might actually end up making the organism stronger in some senses. And it's also it's really challenging to introduce males and have the genetics work out. We don't actually really we don't know enough about the reproduction of this ant in its native range or here, but we do know that when you introduce males between nests, they typically are killed right away. So there's some trick that we don't understand. Could I ask everybody who would to turn on their camera? Because I'm looking at a bunch of phone numbers. Thank you very much. Um,
Put on your videos, guys.
Thank you for your answer, by the way. I appreciate that.
Sure, no problem. Anybody else have a question or thought or?
How fast does this pathogen spread? I mean, you talked about it working most effectively in times of stress when carbohydrates are down, so drought and winter. But how far, you got an infected ant, how far is this ant moving, over time? I mean, how, you showed some rates of spread, but I wasn't clear. This is by individual ants moving or?
Yeah, no, this is the disease moving. So individual ants probably don't move that far, but they do move between nests and they do share food between workers. And then when you sum that process up, you can get really rapid spread rates. So the answer to your question about how fast it spreads is, the short answer is it's variable. In the inoculations that we did in 2017, the ones I showed you the data for, we had spread over the entire field sites we were examining within a year. With everything, the disease had gone from the point of introduction to everywhere within 91 hectares in one case, and in the other case, everywhere within 12 hectares. Every nest that we tested, we'd find the disease in. And that's, but that, in subsequent work that we've done in inoculating other populations, that was a very kind of, I guess, lucky result. The disease can spread rapidly, or it can be a slower process, but typically within 2 or 3 years of getting it to the point where it's locally prevalent, you can go within, say, a 100-meter square area and find it every time you look at a nest. Once you get to that level of local infection intensity, it starts moving pretty rapidly.
Ed, do you want to open up the chat box? I see there's 6 comments there. I don't know what.
Yeah, let me do that. Sorry, I see what it looks like. Okay, great. So let's start at should I start at the top, I guess?
Yeah.
Okay. So then this question, Jim, you already answered this question. You asked this question and I answered it already, right?
That's correct.
And then Peter asks, oh, never mind. There's just a comment. Okay. Kimberly asks, you may have addressed this, but I missed it. Are native harvester ants affected by this pathogen? And then she said to answer my question. Thank you. Yes, they are. Essentially all ant species larger than tawny crazy ants are eliminated typically. The harvester ants hang on a while. In fact, it's a little tough to watch these harvester ant colonies when they're under siege by tawny crazy ants, because what happens is the crazy ants will come every day and try to invade the harvester ant colony, and the harvester ants will get little pebbles and they'll seal up their nest entrance, and they just will shut down and wait. And so because harvester ants can store seed, right? They're seed harvesters. They have a huge amount of food underground, so they can do this for a long time. So every day they open up, every day they shut down, every day they open up, every day they shut down. And in order to close their nests, they can only close their nest from the outside. So every day, a half a dozen workers are there putting pebbles on the outside and sealing themselves out. And then they are, over the period, because harvester ants are big and really heavily armored, over the period of several hours, the crazy ants kill them. So it's kind of heart-wrenching to watch that, but that's what happens.
Leaf cutters. We've had a couple of really giant colonies of leaf cutters, which seem to be dead now.
Yeah. Whether they're dead or not, I actually haven't seen. I've seen leaf cutter. I've not found leaf cutters in populations of tawny crazy ants, so I assume they are eliminated. But those are also big, long-lived colonies. Although their food resource, a fungal garden, is perishable. So I don't know how long they can hold up, underground, if they're like the harvester ants and they've just close the doors.
Yeah, we haven't We haven't seen our leafcutter populations for probably a year and a half, and we had some really good size populations. They were always around.
Well, I'm sorry to hear that. Unless you're a fruit tree aficionado.
Well, yeah, we weren't fond of the leafcutters. That is true.
Yeah, no, they're tough to live with sometimes themselves, but they are amazing. This ant was actually introduced intentionally into Colombia to control in one effort to control snakes and in another effort to control leafcutter ants. And it was done, by a private wealthy landowners. And it turned out to be a real catastrophe for Colombia. But presumably they thought that leafcutters would suffer.
Ed, I had a question on, we know they came from, parts of South America. Have y'all been able to pinpoint where the genetic sort of matchup is between what we have in the US and where the sort of more precise point of origin was in South America?
No, I'd love to do that, but it hasn't been done yet. No, we don't know.
Because I would think that might help in identifying whether the microsporidian is in that population.
I absolutely agree. Yeah, no, I'd love to know that. And targeting that population would be great for a lot of reasons. We don't have that data yet.
And so something like fire ants, these are not super colonial ants. Stupid question, but is a fire ant mound a colony and is different from the mound 50 feet away, or multiple mounds involved in a super colony? Or
yeah, that's a simple question with a really complicated answer, unfortunately. So I'll do my best to give you the synopsis. Fire ants have two social forms that's genetically determined by a chromosomal inversion in the ants. In one social form, the form without the inversion, they're monogyne, and so they have a single queen, a single nest. They make big nests with big workers, and they fight with every other nest. And so that, in that case, that mound is one colony. In the other social form with this chromosomal inversion, they have multiple queens per nest. There is no interspecific, or very, very little, there's no interspecific aggression. And there seem to be few colony boundaries, although that's actually in dispute right now as to the degree to which there are boundaries between nests or not in polygyne fire ants. But the boundaries are very weak. And in fact, under conditions of stress, these colonies will fuse. So they're very weak. And those polygyne fire ants are the ones, when you see these populations where you've got like a fire ant mound every, 4 feet, right? Little ones, just everywhere you look, there's a fire ant mound. That's polygyne fire ants. And the mounds are smaller, the workers are smaller, and the workers are a lot denser. There's a lot more abundant. And they actually seem to be the social form that has the greatest ecological and environmental impacts.
excellent. So what? So what's the process to spread this pathogen? I mean, it seems like we ought to be moving this all over the whole southeast US or something.
Yeah. So it's tricky. I'm doing some of that, right? But there's nobody else doing it. And part of the reason is because you got to know where the infected ants are, and the infected ants are perishable fruit, right? They don't last that long. So there's kind of a fairly intensive effort to keep track of what populations and what their infection status is. And then the inoculations themselves are fairly straightforward, but they're labor intensive. There's no way to culture this disease in the laboratory. You can't, and no one has succeeded with any microsporidian in culturing it outside of its host. So in order to do it, you have to collect nests. Each inoculation is an individual effort of going to an infected population, collecting nests of infected ants, bringing them to the uninfected population, and releasing them in the right way. There are also tricks in that ants, being social organisms, living colonially, have evolved all kinds of defensive behaviors against disease transmission. And so it turns out, although those two sites I showed you, we succeeded right away, our long-term batting average in actually getting an inoculation to establish is closer to about 50%. We're batting about 500. And it takes over a year to know if you succeeded or not in actually getting the disease established, because you're taking infected ants and you're dumping them into this ocean of uninfected ants. And so by the time the disease increases to the point that you can tell if you succeed or not, it takes a long time. So sometimes it happens really quickly, like in those populations I showed you, but actually the rule has been much slower than that. So, and then it takes another 3 or 4 years for the process to play out. So it's not something that is easily commercializable, and organizationally challenging, I guess, to figure out how to ramp it up. So what we've been doing is targeting ecologically sensitive areas, areas with endangered species, areas where the ants are likely to be moved around a lot and are of high value, like state parks. So people take their RVs to state parks, and the RVs get invaded, and then they move the ants somewhere else. And given we have so little public land in Texas, we want to keep it as intact as we can. So that's what we've been doing, and that has occupied most of our bandwidth for doing this kind of work. We just don't have, it's me and 2 other guys doing this. So, and I'm not doing the inoculate, the boots on the ground stuff. I'm doing more research related stuff. So our capacity for work is limited. And I've talked to the USDA folks about trying to ramp this up in other places, and I haven't gotten a lot of traction.
Why not?
I don't know, but I think in part it's because, I showed you that spread, and yeah, there's lots of counties that are invaded, but all of these populations are disjoint, right? They're it's like chicken pox on the landscape. Like, so you happen to live in a population, Gary, so okay, there it's bad, but your neighbors, or you're not your neighbors, but your neighbors' neighbors that don't have them, it's not an issue. So there's not a giant sort of public outcry about it. And the other side is these populations do seem to have a lifespan, right? Because this disease is moving around on its own. We don't understand exactly how, and that's something I'm trying to figure out how to understand. But when we watch these uninfected populations, we might watch this population. I think the oldest was about 16 years old before we saw the disease show up, but then it showed up. So it is a self-limiting problem. So that's been one of the great things that we've learned in this, right? We went from, in 2009, when I started working on this ant, I would go to these populations, I'd see these ants spreading, I'd see these quiet woods with no birds and no insect life, and just, the ground, this seething brown mass. And although the spread was slow, it seemed like it was, everywhere was going to be like this at some point in time, right? And that was, a dreadful position to be in. And what we know now is that's not going to happen. These populations are being moved by people. They get established, they spread, and then during that time period and in that area, you have really severe ecological impacts that are, and for the people who live there, quality of life impacts. But eventually this disease is, even if we aren't moving it around, this disease seems like it's going to find its way to these populations. And and I do have a concern, which is that because these infected populations are perishable and they do seem to decline, they do decline to extinction, it is possible that at the continental scale, we could clear the disease, right? All the infected populations could die. And there could still be a few isolated uninfected populations left that then can spread without constraint. So that is a maybe a science fiction, I don't know, but that is a possible bad trajectory for this.
Is there?
Ed, I've got a question. What's the population dynamics or lack of dynamics in the sort of original area that they were identified in, Pasadena / Houston? Is it still sort of at a super high level, or has there been pockets of it dying out, in sort of its point of origin?
Yeah. So again, in the Houston area, it was like chickenpox, right? They were never connected. There would be a infestation here, and there might, in the Houston area, it was dozens and dozens of these infestations, right? The original Pasadena infestation is gone. That was one of our field sites in our longitudinal study. It acquired the disease and disappeared, at least from the area that we were looking in, and we weren't able to find it in other areas. The prevalence of tawny crazy ants in the Houston area is a lot lower now than it was in 2009. So it seems like there's been more in that area. There's been more population level extinction than there has been population level reproduction. And and something I didn't show data about because I haven't been able to make enough progress on this question, but we have sites where the disease has collapsed and disappeared, and the tawny crazy ants are completely gone as far as we can tell. And then in following and then continuing to follow them to see what happens to the native ants, because another question is what happens, once you have these areas where the tawny crazy ants eliminate all the native ants, and then the tawny crazy ants disappear, what happens with the native ants? Do they come back? How do they come back? What do they look like? And what we have found is that occasionally we'll get these little flare-ups of tawny crazy ants, and we don't know exactly where they come from, but they're universally infected by this microsporidian, and then they die out, and they don't get very big. I mean, it's like it'll be an area the size of my house footprint, say, which I'm going to give away. I live in a little house, 1,000 square feet, and what I believe is going on is that these environments are suffused with spores of this disease, and they're in the soil. I mean, you imagine there's, I don't know if we could count as high as there are spores, and the one way that we can get this disease, the other way besides infected workers that we can transmit this disease, is contaminated environments. And it's a very low rate of transmission, but we can get the disease to transmit if we house crazy ants in environments that were previously occupied by an infected nest fragment. So I think
have
you tried that, or this is just sort of the thought process?
No, we've tried that. That works. It doesn't work at about 40% of the time. That will lead to disease transmission. And so it seems like in these environments that have been occupied by tawny crazy ants, there's a residual resistance to recolonization by tawny crazy ants because of the environmental presence of the disease. That's what I haven't been able to fully test, that statement. But that's the pattern.
The spores would simply be dormant, waiting for the next population to come by, or?
Yeah. And that's essentially what spores are, right? Spores are a way of persisting in the environment when you don't have the conditions you need to grow and reproduce.
So outside of the pathogen, I mean, given how dense the populations are and how they ravage, everything there is to eat, can they simply, overfeed their way into extinction in an area?
Overfeed their way into extinction.
Yeah.
Yeah, so that it's not to extinction, no. That process does happen. So what, I'm trying to figure out how to turn off the chat because I don't think I'm seeing when everybody talks. But okay, I'm gonna stop worrying about that. I don't know how to do it. Okay, yeah. So what does happen is we have areas where the tawny crazy ants have persisted for a very long time. And what happens is you go through this period of extremely high density tawny crazy ants, and then the densities begin to come down, and they come down, and they come down, and they come down, and you end up at this equilibrium state where you have still quite a few tawny crazy ants, but not those kinds of apocalyptic densities I was showing you. And you've got another species of ant, Monomorium minimum, with these very small ants. It's quite successful at competing with tawny crazy ants, and that ant becomes very abundant relative to its natural state. And then that's kind of it. You end up with this kind of two species system of ants, and other insects start to come back to some degree, but it's a very simplified system, and that seems to take about 10 years, just off the cuff guessing. I don't have a lot of populations for which I have that depth of data to be able to sort of quantify it.
I see there's a new question. I think we sort of talked about that from Jim. Do we have data on microsporidian latency?
Latency.
I guess how long it hangs around, or
yeah. So yeah. Yeah, I'm not. If whoever asked that question in the chat, if the microphone works, could they clarify what they mean by latency?
Jim, can you clarify if you're still there?
Yeah, so it sounds like the question is that question of environmental persistence and whether or not environments that had tawny crazy ants remain resistant to tawny crazy ants because of the spores in the environment. And that's still a hypothesis. And no, we don't know how long that spores remain viable in the environment. So, and that's actually something we've been working on actively in the laboratory. It's like almost everything with these microsporidia, it's very challenging. But there are organisms that are really conducive to doing laboratory research, and there are organisms that are not. And unfortunately, this is one that is not. But we are beating our head against that problem.
Thank you.
Yeah, sure.
Oh, well, so boy, I think we're running out of questions, but you have some references up. Is there a handy reading list of literature that you could send our way and I could post online or something? Or
I would be, so everything that is out there, mostly it's stuff that our lab has published. There's been some other labs obviously in the Southeast that have published stuff, but it's all in journals that have paywalls. I'm very happy to distribute those PDFs to whoever wants them. Maybe folks could just reach out to me directly, and then I'll send them on. And if I send them, and Gary, if you get them and you want to pass them on, it's fine with me. So I pass them on to you as well. But you probably shouldn't post them somewhere. You might run into a copyright.
Yeah, right. It'd probably be okay to list titles and authors so someone could self-search this on Google Scholar or something.
Yeah, exactly. Of course, if they don't have the subscriptions, they're just gonna be able to read the abstracts. But happy to pass them along.
Yeah, okay. This has been great. I mean, we've gone a lot longer, but a heck of a discussion. We still have some people. We still have 15 people online. Okay, last chance for questions.
Well, thanks for having me. It's always fun to talk about this stuff and hear people's questions, because it reminds me to think of things that maybe I've stopped thinking about. And it sort of freshens everything up for me. So I appreciate the opportunity and the invitation.